let me set the stage. This may be a little repetitive if you were here last time, but I know there's a lot of new people here. So let me give an introduction to why we're doing this. So I started my scientific journey many lifetimes ago because I wanted to help people. I wanted to use my interest in science and mathematics to understand diseases and common afflictions and health problems for us, for all of us, for humans, and try to understand that and therefore try to develop treatments and therapeutics to improve healthcare and human well-being in general. And that aligns perfectly with Rosalind as a company. This mission, it aligns all of us and it drives us all towards making a really great platform that all of you can use to drive your innovation. So we're here to empower you in this global mission. Okay, so this masterclass. So the highest level, we're here because I want to solidify for you the value of pairing Rosalind with nCounter, the instrumentation and the chemistry. Each on their own, they stand together very strongly, but you unlock the true value and you get the synergy when you combine them together and you extend them from one end to the other. And I'll talk a little bit about this in more depth. At a high level, I want you to understand the principles of the analysis, why we perform each single step in processing of the data and the algorithms themselves. I'm going to be going into math, but don't be intimidated. If you're not mathematically inclined, I'm going to explain it in very basic English that's going to be easy to understand, because I found that those who understand not only the steps to take, but why they're taking it and how it works, they have the best outcomes. If you understand what you're doing and why, you can make the best decisions, get the most accurate analysis, and therefore you have the most confidence to publish your data or perform some validation experiments or to progress your drug development pipeline. So from my perspective, understanding why and how you do every single step is really important to success. So I spoke to this about increasing your confidence and accuracy. And by taking these types of classes, such as this masterclass, you're going to open up new insights you might not have realized. And so that's why we're here at the highest level. If you look at the schedule, this is broken down into 6 sessions. We covered 2 weeks ago, custom pruning and below expressors. Today, we're focused on custom normalization. So normalization is important, as we all know, because you want to make sure you have accurate interpretation of your data. So there's some great best practices that I'm going to talk about, some default methods, but the ability to customize is also important. So now in Rosalind, we have the ability to do some nice customization of your normalization to get the most accuracy in your analysis and the most confidence in your results. So I'll talk about that in a little more depth. But this is the schedule. If you missed our first session, there is a recording available, so please check that out. All of you are here because you are users of nCounter technology. I'm not going to go into all of these points in depth, but after using the technology in the lab for many years and teaching it to a lot of people, there's some key buckets that I've identified as advantages. So I think everything stems from this elegance and the simplicity of the chemistry and instrumentation that makes it very easy to perform and learn in the lab. Very little hands-on time. You get your data very quickly within 24 hours. And then my interest on the data analysis side is that this is a digital technology. So when we start looking at the data, it's single molecule counting, completely enzyme-free, and there's minimal technical variability. So it's really good, high-quality data that's intuitive to interpret. So as we go into some more deeper dives on the data itself, I just want to note some key things here. When we look at the data, again, it's single molecule raw counting. The processing, the algorithms is very fast. You don't need a high compute cluster. You can perform all this on your laptop. The interpretation is intuitive, or it will be intuitive. If it's not yet, I can explain things to you. And this last bullet point is, my motivation for having you all come here today. I want you to understand the algorithms, what they're doing mathematically and conceptually, so you can take control of your data and become the most successful. I kind of laugh as I look at the image of the abacus to the right of this, because to me, it looks like the actual barcodes used by nCounter and NanoString. But for me, it represents like how simple the data analysis is. And everyone should understand what they're doing and why, and you just get better outcomes. Okay, so that being said, in the Rosalind platform, there are different modules to analyze your data. So we'll be seeing some of these in more detail as we progress over the next 5 sessions. Today, we're still in the experimental setup stage. So we'll be looking at how to set up your experiment, how to normalize your data, how to do some nice customizations as well. We are launching this masterclass in conjunction with a lot of great new features for nCounter specifically. I put my heart and soul into these. I directed our team on how to design these, how things should be organized, what features should be included, excluded. So my own personal touch is on all of these features and capabilities that we'll be talking about throughout the course of the masterclass. So I'm at a personal level, I'm just really happy and excited to show all of this to you. But in my experience with NanoString and both Rosalind at this point, Rosalind is the clear solution for nCounter data analysis. It's the most powerful collaborative solution that there is for nCounter data. Before I start jumping into some more technical depth, I want to set the stage at a higher level. So this is the overall workflow for nCounter. So you start in that lab, 15 minutes of pipetting by the bench scientist to set up your overnight hybridization. Come back the next day, you load your samples into the beautiful nCounter instrument, where it's basically automated from there on out for the next 5-6 hours or so. And so from beginning to end, you can go from your samples, your RNA prep, to your data in less than 24 hours. And Rosalind and nCounter work together beautifully because it's a very fast technology to generate your data. You plug that directly into the Rosalind platform, and then you can start benefiting from all of the enhancements, the intelligence capabilities to extend the value of your targeted panel and get the most out of it that you can. So with that stage being set, let's talk about normalization. And so my first slide is this. So I'm going to take a second here. I'm going to challenge you to think about, why are you performing normalization? So I'm going to leave a little bit of silence and just think about why you want to normalize nCounter data. Okay, I'm not going to call on anyone and do any pop quizzes here, but I want to challenge you to think about the basic principles of why we normalize, how we do it, and the choices we make, because the more you understand this, the better your analysis is going to be. I think many people, or some people at least, take this for granted. We just perform normalization because we do, just because that's the way it's done. But we really want to understand exactly what you're doing, or exactly what we're doing. So in my summary, why we perform normalization, can be distilled in this short segment here. We want to adjust the raw data in preparation for downstream biological interpretation, such as differential expression analysis. So that's kind of my highest level, most simplistic reason why we want to perform the normalization. But the question is, what are we adjusting the raw data for? And so we're adjusting for two different sources of variation. There's biological sources of variation, there's technical sources of variation. This is not meant to be a complete list by any means. So there's things on here that I've left out just for space reasons. But biological variation, we can think about differences in the amount of RNA we load per sample. With the nCounter assay, you are loading technically 100 nanograms of total RNA per sample, but there's going to be inaccuracies in pipetting, inaccuracies in quantification. So you want to adjust for that in the data analysis. There's also differences in RNA integrity. You might be working with degraded RNA samples, either intentionally or accidentally, but the normalization process can also adjust for differences in degradation as well. Technical variations we can think about, these are going to be coming from the technology itself. So from sample to sample, lane to lane, or cartridge to cartridge, there can be subtle differences in assay efficiency. We can see those when we look at the positive controls, but this is a source of technical variation that we can adjust for during normalization. One important note before we move on is that you may be thinking about calibration samples for normalization and batch correction, such as during longitudinal studies or from different batches to batches of manufacturing lots. That is not covered in today's session. That'll be a whole other in-depth session by itself. This is outside the scope. What we're focusing on today is those biological variations and technical variations listed on the left-hand side. Just want to make sure that's clear. So let's talk about the normalization, how it's done, the choices we make, and why we do it. So I put together 2 slides on this 2-step normalization procedure for nCounter data. The first step is we want to identify probes to use in normalization. And I listed some best practices here. For nCounter normalization, the best practice has always been normalizing with a certain, let's call it X number of reference genes or housekeeping genes. There's different ways to call them. You may think about other technologies, other strategies. You may be wondering why we don't do a global normalization as an example. And we definitely don't do global normalization with nCounter data because it is a targeted panel. So let's imagine you have an immune-focused panel, and in your particular experiment, you are stimulating with some sort of interleukin or cytokine, something that just stimulates the immune response or the inflammatory response, some sort of stimulation. And so if you look at a targeted nCounter panel, you might have 50% or more of those genes differentially expressed because of the stimulation itself and the focus of the panel. Now, there's a lot of power in that because you're getting the data that you need and you can quickly find the important results. But in terms of normalization, if you did a global normalization in that context where most of the genes are changing, it's not going to be meaningful data. It'll give you a lot of spurious results. So normalization at a global scale, typically, if never recommended for nCounter. So there's this best practice of using a set of reference genes. Now, traditionally, these have been called housekeeping genes. So these are genes you've heard of many times before, GAPDH, beta-actin, maybe even 18S ribosomal RNA. So the thought behind these, it's been decades now, is that these genes, each individually, one at a time, they don't change from cell to cell, tissue to tissue, sample to sample, across different experimental conditions. They're always just going to be constitutively expressed in a constant. So that's why they were, quote unquote, called housekeepers. Over the years, we have learned that a true housekeeper does not really exist. So the preferred terminology now is typically reference genes, and this implies that there is always context. You could identify these stable genes or housekeepers, but they might not be stable at every single context, every single biological situation or experimental condition. So that's why the term reference genes is typically used today. But for practical purposes, I'll be using reference genes and housekeepers interchangeably, but just know there's some history there, and that's why there is some differences. But for the NanoString data, typically the best practice is to normalize with a minimum set of 3 to 5 stably expressed reference genes or housekeepers. If you've been analyzing nCounter data for a while, you know that there's the option to normalize by the positive controls, and these are typically not recommended for normalization. I know perhaps if you've been doing this in NSolver, it's quite straightforward to normalize by the positive controls and then with a set of housekeeping genes. But I would say that the best practice, and this has been established for a while, is not to use the positive controls in normalization. Positive controls can be great for quality control, which we'll talk about in the next session. But conceptually, the positive controls are reflecting that technical source of variation, the sample-to-sample differences in assay efficiency. Now, if you have a good set of housekeeping genes or reference genes, that technical sort of variation, that technical variation that's represented by the positive controls is captured in the expression of the housekeeping genes by themselves. So if you first normalize by positive controls and then normalize by housekeepers, it can be redundant and over-processing. All you need to do is normalize by the housekeeping genes themselves, and that's going to account for both those technical variations and the actual biological variations themselves. So that's the explanation there for why we want to normalize by only housekeeping genes. Now, it's not incorrect to normalize by positive controls and then housekeepers. I would say it's better and it's a best practice to normalize by only housekeeping genes or reference genes. Now, the next question is, how do we identify this set of reference genes that we want to use for normalization? There's different strategies that I'm going to be going into. There's a metric called coefficient of variation. There's an algorithm called GE-norm. This is a whole cottage industry by itself. There's many ways to select what is the most stable set of reference genes. At Rosalind, we select kind of the best practices that have been established over the past, I guess it's been 15 years or so. I can't remember exactly when NanoString first launched. It might have been 2003. So it's been, it's a very well-established technology. And so these are the best practices using %CV and GE-norm to identify your reference genes. So in the Rosalind platform, when we were developing this capability, I wanted to make sure that we had some flexibility. There's no one selection strategy that's going to be 100% perfect in every single situation. There may be edge cases. So if you want to do your own customized selection strategy outside of %CV or outside of GE-norm, we can definitely work with that and get you your nice customization to use in the platform itself. So we employ the best practices that are going to be the most valuable in the vast majority of situations. We acknowledge and recognize the importance of these edge cases, and so we can accommodate those as well. So first step, normalization, identifying the normalization probes. Second step is that you want to then apply your normalization algorithm to the raw data to generate your normalized counts. So it's, kind of a no-brainer. First, you choose your normalization probes, then you apply the normalization algorithm. But in point number 2 here, there's a few key principles that I want to emphasize. The first is that the normalization algorithm, and we use the same algorithm that was established by the NanoString bioinformatics team. We work with them as partners over the past several years. So this is kind of the best practice for normalization. The important principle here and takeaway for you is that normalization is relative. It's not absolute. In other words, the samples are normalized relative to each other. If you have an experiment, you add new samples, or if you take samples out of the experiment, the normalized counts will actually change a little bit. It'll not be dramatic. You might not even notice it unless you're looking at it specifically. But the takeaway here is that the normalization is relative. So adding samples to an experiment, removing samples are going to change underlying normalized counts. So just keep that in mind. It's a good thing to appreciate and understand. It helps you as you design your experiments and analyze your data in the future. Now, when I was building this normalization into the platform and providing my guidance, I wanted to make sure that we could accommodate other normalization strategies as well. Now, even though this normalization algorithm, which I'll be talking about the details of, is the best practice in the vast majority of cases, there's always going to be edge cases that we want to be able to handle and accommodate. So we can actually take normalized data from other methods as well and analyze those in the Rosalind platform. So those are the basics of normalization. I want to show you some numbers, a few equations, some figures, and show you the Rosalind platform. We'll be doing that over the next 20 minutes or so, and we'll, of course, stop for some questions. So that being said, let me switch out of here, and let's go into some kind of practical demonstrations of normalization. So bear with me for 1 minute as I transition over. Okay. So I have some data in Excel. I have opened up some nCounter data in Excel here, and I want to show you some quick demonstrations on some calculations and some data itself. So at the top here, I have this hypothetical experiment containing 5 samples. We know that in each sample, we have a series of negative controls and positive controls. I'll be doing a deep dive on these next time in our QC session. So I won't talk about those much today. And then I'm going to scroll down and show you the data. As I'm scrolling down, I have another question or another challenge for you. How quickly, or is it even possible to recognize if this data is raw data or normalized data? How would you know? Or can you even tell the difference? So as I scroll down, just think about this. Okay, some of you may have noticed something, some of you may have not, depending on your background and your experience level. But quickly, within a second or less, this is obviously raw data. The reason we know this is because if you understand the chemistry and how the technology works, is that it's single molecule counting. So these are integers, 279. So what this represents is 279 counts of the barcode for this ABCF1 gene in sample number 1. So in other words, 279 molecules or 279 transcripts. So when you look at the data, if you don't see any decimal points, if you just see these integers, then it's the raw data. So that just goes to the intuitive nature of the data. It's single molecule counting. You can easily identify the raw data itself. So that was my takeaway from here. Just I want to encourage you, and I'll be talking about this more in the quality control session, but always look at the data themselves. It helps you understand exactly what you're doing and the quality of the data, and it guides you in the analysis. So first step, this is raw data. It's pretty clear just by the nature of the numbers themselves. So we have our raw data. In this case, it's for some housekeeping genes, which we can use for normalization. Down below that, there's some endogenous genes, which are basically our biological targets of interest. These are the ones we're really interested in for pathway analysis and differential expression. So let's talk about how to identify which housekeepers we want to use in the normalization process. I want to talk to you about the most simple case, and that's using that percent coefficient of variation metric that I talked about in the slide just a couple minutes ago. So I want to show you a couple equations, and I don't want to get too math heavy here, but there's a couple concepts I want you to walk away with. So on the left is the formula for standard deviation, and on the right is the formula for the coefficient of variation, or I should say the percent coefficient of variation. So on the left, I don't want to get too math heavy, but the standard deviation is a measure of dispersion. In other words, it looks at any given gene and what is the distribution of the data, what is the dispersion, what is the variation in that gene across all your samples. So conceptually, it's a measure of dispersion for a gene one at a time. It's important to know that standard deviation is meant to look at dispersion of genes individually. It's not meant to be used as a metric to compare the dispersion or the stability of two genes to each other. That's the incorrect application of standard deviation. You get incorrect and false conclusions if you do that. The reason why is that this is primarily driven by the average expression of the gene itself. So it's this term here, the X with the bar over it. That's the primary driver of the standard deviation. I'm going to show you this in a figure in a couple minutes so it makes more sense. But I've seen some incorrect usage of standard deviation in the past out in the field. So I just want to emphasize that standard deviation should not be used as a way to rank order or choose which gene should be used for normalization. What we can use instead is the coefficient of variation on the right-hand side. And this has one subtle change, but it's very important, and that's this mean term at the bottom. So we're taking the standard deviation and we're dividing by the mean. This percent CV is calculated independently for each gene. And the way I think about it is that we're taking the standard deviation and we're just normalizing for its expression level. If we think about the nCounter data, there's 5 to 6 logs of linear dynamic range. So it's a beautiful data set that captures low expressors, medium expressors, and high expressors. And so by normalizing by the mean, we're essentially putting the standard deviation on the same scale. So we can therefore compare it across individual housekeeping genes and then select stable housekeepers. So if this isn't clicking yet, don't worry, I'll show you some figures and hopefully it'll come together in a couple minutes. So let's look at this in more depth. I'm going to go to some more math here. This is the data set we've been looking at. So we have our 5 samples again. I have my housekeeping genes down here, and I've done a few calculations. So I have samples 1 through 5. Right now, they're sorted alphabetically. Let me sort these by standard deviation. So just sort the data here. And at the top, I have my gene CC2D1B. It has the lowest standard deviation and a rank order of 1, meaning it has the lowest standard deviation. And if I scroll down, you'll see that the SDs increase as well as the rank order. So the lower the rank order here, the lower the standard deviation. And so this is just ordered from low to high. Okay, and I've done the same thing, but I've done the coefficient of variation calculation. Remember, this is simply just the standard deviation essentially normalized by its expression level. So we can account for that large dynamic range of the assay and then compare the stability of these housekeeping genes to each other. So let me let me sort this table by percent CV by doing this. And now we have the rank ordering with 1 being the lowest or the most stable according to CV, and at the very bottom is the highest. So important thing to note here is, the lowest or the most stable gene by percent CV is rank order 1. But if we look at standard deviation, it's actually 7. Another thing that popped out to me is that this gene here, whatever it is, CC2D1B, when we look at its percent CV ranking, it's 26, so not very stable, but it's actually number 1 by standard deviation. So I think you might be starting to understand why standard deviation isn't a useful metric. And let me show you a couple figures to drive this home, kind of bring this to life here. So what I plotted was at the top, Each data point here is a gene, and its average expression is on the x-axis, its standard deviation on the y-axis, and this is the relationship that holds true. The higher the average expression, the higher the standard deviation. This is just the way it is mathematically. So there's no point in trying to select housekeeping genes by standard deviation. Now let's perform the next step here, perform that coefficient of variation calculation, essentially normalizing by average expression, and that's shown here at the bottom. So quickly what you can see is that this really high expressor in this data set actually has pretty average percent coefficient of variation. And when we look at the actual housekeeping genes themselves, most of them are in this middle range of expression. There's a certain ones that have high percent CV that we would not want to use in normalization. And we want to select some of these down here for normalization because they're very stable. In other words, they have low percent coefficient of variation. So if you haven't seen this type of plots before, it's very basic, but it can be skipped over just to kind of rush to get through the data analysis. But it's a very good principle to understand. It'll serve you forward in many different technologies beyond nCounter. So that's the relationship between SD and percent CV. Clearly, we want to use coefficient of variation for our rank ordering. So all that conceptual stuff being explained, what we want to do is then select which housekeepers we want to use for normalization. So in the traditional method, you might rank order these housekeeping genes by percent CV and maybe take the top, let's say the top 10 that have the lowest percent CV and use those in the normalization. That's a valid approach that's been used for more than a decade now. It's a really great way to go. There's been some advancements that I want to talk about, and that's the next or the alternative way to identify these sets of normalization genes. And that's through an algorithm called GE-norm. This was first identified or created for qRT-PCR data. Wow, 2 decades ago now. I remember this being new and really exciting when I first read it. This algorithm was first developed for RT-qPCR by Joe van de Sompele. He's kind of a luminary in this field. This is a really big active area of research. You may or may not have read articles about how to choose the best normalization genes or normalization strategies for gene expression in general, but he's a really big name in this field. So I want to pull out a few quotations from him where he was looking at the challenges with normalizing qPCR data, RT-qPCR data, and what he observed and why he wanted to develop this algorithm called GE-norm. And this is the algorithm that nCounter Advanced Analysis and Rosalind uses. So the first statement here is that first, it's just generally accepted that you should be normalizing your data. And again, this is for qPCR, but it holds true for NanoString. That you should be normalizing your data by a stable internal control gene or reference gene. Okay, so we've already talked about that. That makes sense. But now he starts talking about this conundrum that we find ourselves in. However, in order to validate that the gene is stable, we need to have prior knowledge that is actually stable. So it sets up, I'm not going to kind of read this to you, but it sets up this, what he calls a circular problem, or maybe a catch-22, in order to identify from our raw data which genes are stable. We need to have a good normalized data set in order to identify the stable normalization genes. But we can't generate that normalized data set without first having a raw data set and then identifying stable genes there. So it's just this circular problem. Like, how do you get into this solution if all you have is raw data, but you need to interpret the normalized data in order to find stable genes? So this is kind of the underlying problem. You may or may not have thought about this, but this is the genesis of why this type of algorithm was created. So what the author did, he created GE-norm, and he created this based off of a very simple and very ingenious principle, is that if there's any two genes that are both stable and good for reference gene normalization, then they should have the same ratio between each other in every single sample. So this right here is the main principle of the algorithm. I want to show you a figure of what this might look like. But this is the principle behind how you identify stable reference genes using GE-norm, and it's the fact that they have this stable ratio across all your different samples. So if that's not making sense yet, let me show you a couple figures. What I have to the right is our same set of data. This is our housekeeping data. And I took a few of these housekeeping genes. And remember, this is still the raw data. And I created a couple figures. So on the right here, this top figure, it's these sets of housekeeping genes, and then what is its raw counts in our samples 1 through 5. And you can see here a few different patterns. There's this gene at the very bottom, GPatch3, relatively consistent across the different samples. There's this gene in this blue here, this aqua blue AGK. You can see it spikes in sample 4. There's this one at the top, which is, I believe, FCF1, where it has a different pattern where it goes down, then up, then down. So there's all these different patterns of reference genes across your different samples. And how on earth do you find out what should be used for normalization? So we can apply the GE norm algorithm to do that in a very principled way. Now, the way that it's done mathematically is going to be beyond the scope of this call. I'm not going to go into the math of the algorithm. It's going to be more complicated than what I'm showing you. But conceptually, I want you, again, to think about the ratios between sets of genes. So that's what I have plotted here at the bottom. So I took 3 different genes. So I took SF3A3, ZNF143, and HPR-T1. So these dark blue bars is the ratio in raw expression between SF3A3 and ZNF143. And so in sample number 1, we see this ratio is, roughly 2, about the same in sample 2, about the same in sample 3, 4, and 5. So this ratio between these 2 genes is relatively the same across all 5 samples. Therefore, these 2 genes may be stable and useful for robust normalization. Let's take a look at another pair, SF3A3 and HPR-T1 in the orange. So it's a little above 1 in sample 1, 2.5 or so in sample 2, sample 3 is roughly 2, and then sample 4 is very different. In this case, the ratio flips. And now in this case, the expression of HPRT is actually greater than SF3A3, giving us this negative ratio. But then it's positive again here in sample number 5. So clearly the ratio between these 2 genes, SF3A3 and HPR-T1, it's not stable. It has this abnormality or this aberration in sample 4. So clearly using these 2 genes in conjunction is not going to be a robust normalization strategy. And as one additional example, let's look at ZNF143 and HPR-T1. We can see in sample 1, this ratio is roughly -2, then it's positive at sample 2, then negative in sample 3, then very negative in sample 4, -5 even, and then around -3 in sample 5. So this pair of genes is not stable either. So what the GE norm algorithm does, it looks at all of the possible pairwise combinations of all housekeeping genes in your panel and pulls out what is the most stable set of housekeeping genes, simply by looking at the stability of these ratios across all combinations. So it's conceptually, that's what's happening. Mathematically, it's a little more involved than what I've explained, but hopefully this gives you a sense of what we're doing with GE norm and why it's a good tool to use for normalization and why it's been the default, the gold standard for nCounter normalization and qPCR normalization for the past couple decades. Okay, so let's imagine that we've selected our best set of reference genes or housekeeping genes for normalization, either by %CV or by GE norm. And how do we now normalize our data? That's meant to be shown here. And again, I want to emphasize that the best practice is to normalize with a minimum set of 3 to 5 stable reference genes or housekeeping genes. The reason why, I want to show you a brief anecdote on the right-hand side, because I haven't forgotten about this since I read it many, many, many years ago. It's just really eye-opening if you haven't seen this data. This is RT-qPCR data. They're looking at the gene OGG1 in normal controls and some gestational diabetes patients. This is human placenta samples. And so what's shown here is that you can normalize OGG1 by individual housekeeping genes, and you get a completely different result depending on the gene you normalize with. If you normalize OGG1 with 18S, your conclusion is that the gene is lower in diabetes. If you normalize by beta-actin, your conclusion is that the gene is higher in diabetes. If you normalize by GAPDH, your conclusion is that there's no difference. So there's great risk in normalizing by an individual reference gene or housekeeping gene. This should never be done, in my opinion. It's really risky just because of this type of phenomenon. Down below, it's a little more depth than I might want to get into now. This is basically the authors employing the GE norm algorithm, which is the one I just showed you, and they use it to identify, okay, what is this nice set of stable housekeeping genes to use for normalization? And that's shown here on the right. The main conclusion of this analysis is that there was no difference in this gene between control and diabetes, but depending on your normalization strategy, you might get the opposite result. So it was a really interesting paper. I'll never forget about it. So that being said, that was a little bit of an aside. Let's go back to our normalization calculations. So for the sake of argument, let's pretend that I have identified this set of genes to use in normalization. To calculate our normalization scaling factors, we perform 3 steps. And the first step is represented here. We calculate the geometric mean of this set of housekeeping genes in each sample individually. So this number, if you want to look at my Excel formula, it's simply the geometric mean of all of these housekeeping genes in this sample. Similarly, this number 155 is the geometric mean of the same set of housekeepers. You go to the next sample, geometric mean, so on and so forth. So it's just the geometric mean of the selected housekeepers. Then what we do, we calculate this global geometric mean. So we're taking all of these values, all of these sample-specific housekeeping geometric means, and calculating the global geometric mean. So this is 212. Now there's a nuance here I want to point out, is that this could be done differently depending on the software you may be using. In Rosalind and nCounter Advanced Analysis, calculation of this global mean is done by the geometric mean calculation. In nSolver Basic, this is actually done by arithmetic mean. So I would suggest, as a best practice, you always do this with the geometric mean. It's going to be preferred by statisticians, and it's typically the best practice to do it. There's going to be very little difference in actuality when you look at arithmetic versus geometric, but as a best practice, I would certainly always use the geometric. That's going to be the preferred way. So that's the second step here, calculating this global mean. And then finally, you calculate your normalization factors. So that's just the ratio between your housekeeping gene geomeans and this global geomean. So for sample number 1 here, our normalization factor is 1.26. That is calculated as the ratio between our global geomean and our individual sample geomean. So 212, 196, that gets us 1.26, the ratio. So just to be a little more explicit here, sample number 2, it's the ratio between our global mean and our individual sample mean, and that gives us our value of 1.37. Here, again, it's the ratio between our global mean and individual sample, and we do that for every single sample in our experiment. So I want to explain this a little bit in more depth, because we will see these again. These normalization factors are actually used for quality control. Conceptually, what we're calculating here is the relationship between an individual sample, or I should say, the relationship between the geometric mean of the housekeepers of an individual sample versus the average across the whole data set. That's essentially what we're looking at here. So what 1.26 here represents is that this sample, the difference between the geomean of the housekeepers in this sample and the average of all samples is 1.26. This sample happens to have the same value as the average, and then the relationship between this sample, which is sample number 5, versus the global geomean of the housekeepers is about 0.89. So conceptually, these are just distances or ratios or relationships of individual samples to the global geomean of the housekeepers. So we'll see this again in quality control, which I'll talk about it in the next session. So we're going to be using these normalization factors to normalize our data. Let's look at how that's done next. So we want to take these normalization factors, apply them to the raw counts, and we generate our normalized data. So that's shown here in the next tab. Let me scroll down to my data. These are the same calculations. I have my sample-specific normalization factors, and then we normalize following this procedure here. So we've already done number 1. We've selected the reference genes. We've done number 2. We've calculated the sample-specific normalization factors. That's what these things are here. Sample 3, we multiply the raw data by their corresponding normalization factors. So that's shown below. So we take sample number 1, the counts, raw counts for A2M, multiply this by this value here, the sample normalization factor, and that gets us this value here. So let me just make that a little more clear. We multiply the raw counts of this gene, sample number 1, versus the normalization factor for this sample, 4585 multiplied by 1.26 is this value shown in this cell here. Let me be a little more clear with the next example. We take sample number 2, we'll take its raw counts, multiplied by the normalization factor. That's how we get this value. Sample 3, I think you probably get the idea. Take this raw count, multiply by this normalization factor, we get this. One more time, sample 4, we take this raw count, multiply by the normalization factor, so on and so forth. And we do this for every single individual gene, or about 800 genes in this particular panel. So that's step number 3. Final step is log2 transform of the data. So for various reasons, it's statistically preferred to perform all differential expression and statistical calculations on data that has been log2 normalized. So that's the final step. We convert your linear raw data to this linear normalized data. Then we log2 transform it, which is shown here on the right. So I simply took the log2 value, or I applied log2 transformation to all of these values here, and that's what's shown on the right-hand side under log2 normalized data. This is the input to differential expression, pathway analysis, cell profiling, etc. So that's normalization in a nutshell. Looks like we have about 11 minutes left. I'm going to show you a couple things in the Rosalind platform, and then we'll stop for some questions. And I've probably been logged out by this point, so let me log back in. And go to what the normalization screen looks like in the platform. So if you're brand new to Rosalind, this is what it looks like the first time you log in, or this is your home navigation screen, I should say. I have a folder created for my nCounter gene expression data. I'm going to go to that and go to an experiment I've already created. So I've imported some RCCs. I'm in the middle of setting up my experiment. So let's take a look at what that looks like if you're in the middle of setting up an nCounter gene expression experiment. You eventually get to this screen here after you upload your RCC files. And there's 2 paths. There's this default path and advanced path. The default path is the path that's been available historically for the past several years within the Rosalind platform. All nCounter users have gone through the default path. What's new as of a couple months ago is this advanced path. The advanced path allows you to access customized low expression or customized pruning of low expressors, which we talked about last session, and also customized normalization. That's accessible for the advanced path or this advanced workflow. The advanced workflow is available to our paid subscribers. So all of our academic researchers, our academic subscribers, professional enterprise subscribers can access the advanced path. If you have a sponsored subscription via NanoString, you have option to the default path. So if you do want to access the advanced paths, you will need to upgrade your subscription. So let's take a look at the advanced paths and the advantages of having this available. So this is custom pruning screen, which we talked about last time. I'm not going to go into depth, but if you weren't here last time, I made one important point, and that is the steps or the order in which we process the data. So historically, in other software packages, you have selected your housekeeping genes for normalization first, and then you've pruned the low expressors. What that can result in is that you can actually have some housekeeping genes below background or below your negative controls enter the normalization process, which clearly you want to avoid because you're just introducing noise into your normalization, and that just cascades downstream, adding noise to all of your interpretation. So when I was thinking about designing Rosalind from scratch, I wanted to challenge the status quo. I didn't want to take anything for granted, and I thought about this step, and I told the team, let's reverse the order. We want pruning to come before housekeeping gene normalization so we can identify those housekeepers that are below the negative controls, get them out of the data set, remove them so there's no danger they're ever going to be used in normalization. So that's a really important point here that you don't realize. It's not very obvious, but we put a lot of thought into it. Custom pruning of low expressors should come before normalization gene selection. If not, there's going to be noise introduced into your normalization. So I'm not going to go into this in depth. Again, there's a recording to talk about this. But after you prune your low expressors, the next step is to come to normalization. So we apply the GE norm algorithm, like the one I just showed a few minutes ago in Excel. And what the output of GE norm looks like are all of these genes highlighted in green. So you can see recommended probes for normalization, 8 of them. These are the 8 most stable genes identified by GE norm. Those are going to be by here, on the right-hand side by default. These are the ones we're going to recommend by the GE norm algorithm. In the vast majority of cases, this is going to be the way to go. It's going to be a robust normalization. If you don't know how you want to normalize coming into the experiment, use GE norm. It's going to give you the most robust results. But there's a lot of flexibility here as well. So this table in the middle is interactive. You can sort, let's say, by category. So let me sort by category. And you'll notice there's positive controls here, housekeeping genes, as well as endogenous down at the bottom. So typically when you're normalizing nCounter data, you are going to use housekeeping genes. That's the best practice. That's going to give you the most robust results. As I mentioned in the beginning, you typically don't want to normalize by the positive controls, but we did include them then here because there might be edge cases where you want to use them. For example, if you're working in a diagnostic or clinical setting and you're performing an analytical validation of your actual panel itself, you want to perform a linear dilution series, let's say, 500 nanograms, 100 nanograms, 50, 25, 10, 5 nanograms of input with the exact same sample, the same panel. You want to look at the linear dynamic range and sensitivity. In that case, if you normalize by housekeeping genes, you are going to actually cancel out the effect of the linear dilution. So in that situation, positive control normalization is probably going to be best for you. So again, there's edge cases for positive controls, which is why we included it here. You can pick and choose housekeeping genes as well. So my recommendation in my experience is that if you're in a discovery mode, typically, maybe this is a new academic project you're working on, maybe you're working in preclinical discovery or some discovery setting at an institution, you might not know, or you probably don't know how to normalize the data. So in that setting, in that situation, using the GE norm recommendation is the way to go. However, once you progress, let's say in your drug development, maybe identify some candidates, you want to get a little more strict and start locking things down, then at that stage, your team, perhaps it's your bioinformatics team, will actually lock down what is the best set of normalization genes to use. So in that situation, they're going to tell you, you need to normalize by this set of 10 housekeeping genes at every single experiment. And so you can do that with Rosalind. So if they want you to add DDX50 in every single experiment, then you can add it to the normalization by doing that. Or if they also want you to add COG7, you can add it to make sure you get the exact same normalization every time. The reason I'm pointing this out is that GE norm is empirical. So it just takes a look at your data and selects what is the most stable set looking at the data specifically. If you change the data set, you add samples, remove samples, the recommended genes by GE norm might be a little different. So there might be a little change from experiment to experiment. That's why we allow you to do this customization. So you can lock down your normalization so it's exactly the same in every single situation. Okay, let's see. What else do I want to recommend here? I know we can pause for some questions. Let me point out kind of the last thing I want to talk about is this export raw data here. So if you click this button, it allows you to download a spreadsheet of the raw data. And then if you have an alternative approach to normalization, I talked to some researchers recently who like to look at correlation of housekeeping genes. And they really like to first filter by average expression of their housekeeping genes, then coefficient of variation, and then look at which housekeepers have the best correlation, pairwise correlation to each other. So you can do that type of analysis by exporting the raw data, performing that customized approach outside of the platform, and then coming back in to select those housekeeping genes you want to use. So there is a lot of flexibility here. We do employ the best practices that have been well established for more than a decade now of %CD and GE norm. But of course, you can use your own approach, export the raw data here, perform those calculations, come back in here and apply your normalization. So I know there's 3 minutes left. You might have some questions, so I'll pause. Tim, if there's any questions I can address, or Kuki, I'm happy to take those now.
There are some here. So Kuki, I'll take the first one if you want to prep on the next one. And let's see here. Let's get started. So the first one was, thank you all so much for the questions. During normalization, how can we indicate NTC as low count as normalization value doesn't justify that one?
Um, I might need a clarification on that, but I believe it's a reference to an NTC or a no template control. So that's, I think the analogous thing to that in the NanoString assay would be like a water lane, where you're loading water instead of an actual RNA sample. So you look at your sample and all the values might be 1 or 10 or background levels. So typically you would not want to use that in your normalization. So you would just exclude that sample during the normalization process. And again, it's because the normalization is relative. So it's all relative to each other. So if you include that water lane or that NTC lane in your normalization, that kind of technical sample can actually impact the biological samples, and you just take it out of your experiment before you perform normalization.
And by the way, it looks like one of the questions did get clarified and resubmitted. So thank you so much for doing that. I'll go ahead and read this one. This one was, I got a little confused. As far as I understand, we can compare the expression of a gene across the samples, but not the expression of genes in a sample of nCounter data. Could you explain why we can use counts from different genes to calculate the normalization factors?
Let me think about that one. So yeah, within any given sample, so I'm talking about sample number 1, the way that the nCounter data is structured is that you can't necessarily compare in a quantitative way what is the expression of gene A versus gene B. That's just not how the nCounter data works. The nCounter data is relative across different samples. It's not used to compare the relative expression of 2 genes in the same sample. So when we're talking about housekeeping gene normalization, we are kind of taking the average of a set of housekeeping genes in each sample, but we're not necessarily inferring what is like the difference between each individual housekeeping genes in terms of differential expression. It's just an average value to get an estimate of essentially how much RNA is there in the sample or what is the technical variation there. So I'm not sure if I'm clearly answering that question. So happy to address that a little further, but that would be my response.
Thanks, Ryan. Let's keep going. And as always, and I appreciate the folks are updating their questions as we go. So if you want to clarify, if Ryan's not giving you additional detail you'd like, you have 2 options. One, you can clarify that question now, or you can also follow up with us. And we'll post on the chat the ways that you can reach out to Ryan's team and ask more questions. I don't think we clarified also at the beginning, as the lead of our customer experience team, Ryan's team is dedicated to supporting typically our professional and enterprise customers. So we're doing a lot to work with everyone right now on the transitions in NanoString nCounter subscriptions. So anyway, Ryan's happy to answer questions and help you all through all of this. That said, I want to ask the next question was, and this I like this one too because it talks to some of the historical aspect of what folks have been working with. So this question says, I used stepwise normalization with GEnorm 2 and nSolver version v0.4 by NanoString before. Is this okay? And of course, Ryan is super aware of the different versioning and the different capabilities of some of the other software NanoString's provided. So with that, I'll turn this one over to you, Ryan.
Yeah, I think there's always nuance here, and the devil's always in the details. So hard to give just a blanket answer, but I think the GEnorm algorithm is the same. So it's the same package, the same algorithm itself. So the identification of housekeeping genes in nSolver is the same, or nSolver advanced analysis is the same as in Rosalind. So if that's how the housekeepers were selected, it should be okay. There was one nuance. So this is advanced analysis. I got it. Okay. So yeah, I would say the selection should be the same in nSolver advanced analysis as well as within Rosalind. So it should be good. The difference is if you did it in advanced analysis, there's that issue I mentioned in the order of how we perform pruning versus selection of housekeeping genes. So in Rosalind, we first prune all of the genes, and in that step, we can get rid of housekeeping genes that are below the negative controls. And then once we do that, then we can apply GE norm to identify the most stable set of housekeepers. In advanced analysis, it's going to be reversed. First, you apply GE norm to identify housekeeping genes. Then you prune the low expressors. So in that order, there is the chance that you could have some low expressors below background enter the normalization process. So I don't know if that happened in your case. It doesn't always happen, but it's something that comes to mind that there's a potential that could have happened. But the algorithm itself is the same.
So I think I'll make one other generic comment. There's, as Ryan tells me, there are a lot of things you can do in nSolver or in the separate advanced analysis, but sometimes it's not the right thing to do. And I can assure you all that when we were guiding the development of these advanced capabilities, Ryan and his team, with their deep expertise in NanoString data analysis, was absolutely adamant about how we had to make sure Rosalind guided you to being able to get the best answers. So that you can be sure looking at that data even again as a Rosalind analysis will generate great results for you. The next question was, your Excel sheet template is very helpful. I have a multi-merge experiment where I had to do a manual normalization combining 2 custom gene panels for a non-model species. And even though I worked with NanoString support, I did not go through all those steps, but I would like to. Would it be possible to get a copy of your template?
Definitely. Yeah, and we did this same thing for the first session with pruning. I made a nice Excel spreadsheet showing calculations. Happy to share the same one as well. So that should be available on Rosalind Academy or on our blog as soon as we're able to post it. So 100% yes.
Thank you for that. The next one, can you go over some reasons why to use log2 normalized data versus normalized data?
Yeah, primarily it's a statistical thing. So let's take a t-test as an example. So if we want to run differential expression with a t-test, and I'm not advocating that we do this, but just for argument's sake, a t-test, the assumption there is that the data are normally distributed. And so when we talk about kind of the raw data of NanoString, it doesn't necessarily conform to that normal distribution, so it might not be valid to apply that t-test to this non-normal distribution. Statistically, after you apply this log2 transformation to a raw data set like an nCounter data, it does become more normal-like. The distribution kind of shifts to become more normal, and therefore it's more applicable to apply a test like a t-test. So long story short is that there's various statistical reasons why log2 data is preferred, and it's purely a statistical thing. It changes the distribution of data, making it more amenable to statistical inferences.
Perfect. Next, is there a way to include panel standard normalization and to use the normalization factor to normalize the raw data?
Not yet. Not yet. This is part of what I'm working with the team on. Panel standard normalization is not yet available in the platform. I could actually show you in my version here. I have a preview of what it's going to look like. So it is in development. Panel standard normalization, we recognize the value. So if you need to do that type of analysis now, the great thing you can do is do the panel standard normalization in nSolver, export the raw data, and then take it directly into the Rosalind platform. But I do acknowledge the importance of the panel standard normalization, and that is in our development pipeline.
Okay, thank you, Ryan. The next one, for NanoString microRNA panels, if the housekeeping genes, which are mRNA, have a flag for low counts, does it necessarily mean that the microRNA data might not be accurate, since normalization is performed by the top 100 microRNAs?
It depends. So that's going to be a lot of my answers here. It depends on the data itself. But microRNA is definitely a different strategy as the questionnaire indicated. So there's a strategy called top 100 microRNA normalization, where you're not actually normalizing by mRNAs. So a lot of the importance here is going to be the sample type. So if you're using cells or tissues as your sample type and extracting RNA from there, the biology is such that you might get, let's say, hundreds of microRNAs that are expressed. So, 200, 300 microRNAs, 400, 500 or more that are expressed. And since you are extracting from this cellular context of tissues or cells, you are going to get this expression of mRNA genes that the panel can detect. If you're using biofluids, it's a very different situation. It's typically some sort of cell-free preparation from blood, serum, urine, something like that. So you might not necessarily have mRNAs present in that sample type. So if that is your sample type, you don't have mRNAs present, not necessarily a bad thing. It just depends on the biological context.
Okay, I think this is the last question, Ryan, and it's an excellent segue and a reminder for you to grab the session schedule and bring that up for the close. The last question is, are there any sessions on meta-analysis coming up?
So in 2 weeks, it's quality control. And then I've organized this just in terms of just general processing steps, kind of walking you through from beginning to end. And meta-analysis is we're saving the best for last. So that is on June 27th.
But of course, if you have questions on meta and you need help before that time, please reach out. And Cookie, if you could provide in the chat the best address folks should be using, whether it's the customer success one or another, please make sure that's posted. And thank you all so much for the question. So Ryan, I'm going to hand it to you for your close. I'll just simply say thank you all for your time, your attention, your passion for discovery, and for trusting the Rosalind platform to help you get there. I hope you enjoy these sessions, and I will turn it over to Ryan.
I will echo those comments. Yeah, thanks everyone for joining. I was really happy to see the turnout. And thanks for all the questions and the engagement. Definitely want to share my knowledge with all of you and help you be successful. Hope to see you in the next session coming up, quality control. Our attendance and registrations keep increasing over time, so this is really great to see. In the meantime, before May 16th, if you have questions, please reach out to our customer success and support teams. If not, we will see you on May 16th. Thank you.